and I've been working on the Conservation Canines program since 2009, so I've gotten the privilege of driving Tucker around. I say I'm his chauffeur since 2009, and I've gotten to work with Liz Seeley since 2010, and I'll let her introduce herself.
Hi, I'm Elizabeth Seeley, you can call me Liz, and I've been with Conservation Canines since 2008, working on a variety of studies all over the world and all over the U.S. And currently, I am the Sound Watch program coordinator, so if anybody wants to get out on the boat this week, we have a couple extra spots for volunteers. But my main passion is Conservation Canines and working with Scat detection dogs. So without further ado, I know we're a little behind. We'll just start talking about our southern resident killer whale study through the Center for Conservation Biology. Sam Wasser's been working with that since 2006, and we've been working with Scat detection dogs.
So I actually haven't had an opportunity to be here too much this year. Lots going on the west side. So I'm not sure how much you guys have gotten on the background of the southern residents. So just one slide really quickly. 3 pods, J, K, and L, make up the clan, the one clan of the southern residents, designated as a distinct population segment of the worldwide Orcinus orca genus species. They were listed in 2005 when there were 88 animals, and now sadly we only have 83 animals left in the population, so it's actually going the wrong way. They were listed in 2005, as I said, after a long time, several years of the government calling for studies to try and understand what was causing the decline, and the 3 main threats that were identified were toxins in the water. So we'll get into the specifics in a couple slides. Vessel presence and associated noise, so that's everything from your incredibly large container ships, commercial fishing, commercial whale watching, private recreation and whale watching. Pretty much anything with a motor makes noise. And then the lack of prey, so lack of quality and quantity of prey. Specifically, Chinook salmon studies have shown them to be obligate fish eaters, very, very honed in on the salmonid species, and within that, very, very preferentially targeting the Chinook salmon, the king salmon, which makes sense from an evolutionary standpoint because those are the fattiest fish, so the highest lipid content still are, but certainly in the past were the Chinook. So why Scat? Why study Scat? So what we say on the boat, it's not very professional, but it's the mother lode. Like it's a gold mine. When you get a good Scat sample, we just can't help but getting really excited about it because we can tell so much about it. Individual, so who was the whale that pooped it out? What is their sex? Even if we don't have their DNA on file, which most of the whales have been DNA typed, but even if that particular whale, say it's a calf, we can tell the sex. We can tell if a female is ovulating, we can tell if she's pregnant, we can tell if she's pregnant, where in her pregnancy she is, so is she early term or late term pregnancy. Nutritional status, so how much are the whales getting to eat and how nutritious is the food that they're getting. And then physiological and disease state. So really, the list is actually longer than that, but you can get an idea from this list just exactly how rich of a data set that is when we can find it. So how do we find it?
So that's where I come in, and we find it using Conservation Canines. They are these highly trained Scat detection dogs, and they have been part of the Center for Conservation Biology since 1998, actually. And there's a couple of things they look at in order to get a conservation canine. The first is that all of our dogs are rescue dogs. They're either from animal shelters or owner surrendered. And the point is, they're the dogs that people can't adopt. Like it's a normal family, if they're looking at this dog, it's crazy, it's really energetic, it's obsessed with a toy. All of our dogs have this play drive that is just unbelievable. If you throw their toy into a bowl of food, they're not going to eat the food, they want to play with the toy. And then they're like, oh, there's food there, and they really contemplate whether or not they're going to drop the toy to eat the food. So those are the dogs that families regularly can adopt, and they're either at a kill shelter, which is where a lot of our dogs come from, or they just stay in the shelter for many years. And then shelters, when they hear about our program, they start contacting us, and they're like, we have this dog. So we say, okay, send us videos. And we're looking for dogs where if you throw the ball, the dog, you know how a tennis ball just bounce. We look for the dogs that just go like this too. And their heads just bounce up and down with the tennis ball until they actually get it in their mouth. And I actually have one at home where she has one in her mouth and she'll still watch another tennis ball. And then if you throw another one, she'll try and drop it and pick up two and then still watch the other one. So we're looking for the crazy, crazy dogs. We have working dog breeds anywhere from Jack Russell Terriers to Husky mixes, Border Collies, pretty much anything you can think of. We have blue heelers, black labs, you name it. They're all that kind of high energy, crazy, obsessive breeds. We don't usually go for the mellow ones like a golden retriever or Great Danes or hounds actually. We want dogs that will sniff the air. In particular, for an example, hound dogs, when they're hunting, they actually track animals from sloughed off skin cells left on the ground. So we chose the ones that'll sniff out the air instead. Just to give you a brief idea, that's Tucker on the right-hand side, for those of you who don't know him. He's been a conservation canine since 2006. The killer whale work and the Alberta oil sands work up in northern Alberta in Conklin, actually, is his first two studies. And then this one down here on the bottom left, his name is Dio. He's a blue heeler, and he is the current whale dog in training. So he will be Tucker's replacement when we go full-time again next year. All right, so we have our facility in Pack Forest, Eatonville. It's right in the foothills of Mount Rainier. We couldn't ask for a more beautiful setting if we tried. We have this gorgeous barn that can house up to 32 dogs. They have indoor, outdoor kennels. We have a huge fenced yard that we use for break time, and an itty-bitty yard that we use for potty breaks only, where we're like in and out, let's get it done, it's mealtime. And then we have over 4,000 acres of forested land that we train in, and the dogs get exercise through there regularly, whether it's jogs or bike rides or hikes. And then on top of that, we do all of our field exercises out in the woods before we go on a study. We have dogs literally all over the US at any given point in time, and we have dogs that have gone internationally. Tucker has gone to St. Lucia. He and I worked with arboreal iguanas down there. We've also gone to Cambodia to look for tigers and leopards. We had a dog team go to Africa for lions and leopards and cheetahs down there. So we have dogs that travel all over the place for many, many hours on a plane. And we also have teams currently even in the US working in Oregon for pine marten, New York for moose. We have a couple different teams on wind farms looking for bat carcasses and trying to figure out the impact of that. Our map is just cluttered with waypoints. So if you guys ever wanted to learn more information, you should look us up on Facebook, because we post photos all the time of our super cute dogs and the studies we're working on. And then obviously we're down here on a boat, sorry, should have mentioned that, working with the killer whales out here. So if you ever see our little boat running around, that's us with Tucker. So the other question people come up with, usually on a daily basis to me, is how do you train a scat detection dog, especially to find whale poop, especially when it sinks? And then the next question is, it floats? And I'm like, oh, okay, yes. So whale poop floats anywhere from 5 seconds to about 45 minutes, maybe, on a calm day with a real fatty sample. And the way you start training a dog is you start in a room where there's no other scents, no other distractions. They just have this box, which is the box training on the top left. And you just put out mason jars and you have one jar with the poop in it. So it's the only thing the dog can focus on. You take them down under control. As soon as the nose hits the poop, you throw the ball. And it doesn't have to be when the dog's looking at you. You want the dog solely focused on that poop when it sniffs when it sees the ball across its field of view. And that's an immediate connection for that dog. So then he automatically starts to realize every time he smells that poop, he's gonna get the ball. You just do it over and over again until as he's got like one nostril on the poop, the other eyeball is like looking up at you for the ball. And at that point, you can move on to the next exercise. And that's where the sit exercise comes into play. So then you move it outside, and pretty much the whole thing about training is repetition. So outside, you do exactly the same thing. You set up a row of samples. You let the dog roam around in the grass. There's all these other things it has to think of, like there's a wind, the grass smells like other dogs, it smells like people, it smells like, I don't know, 2 different shoes or whatever they're smelling. And then all of a sudden, its nose ends up on the poop it's looking for. And you throw the ball across their face again. They're like, oh yeah, I get to play when I find this, that's so cool. And then you do it over and over again, and before you know it, they're sniffing a poop and looking at you like, okay, give me the ball. And you also have them sit. So once they're looking at you, you can be like, sit, and throw them a ball. They're like, yay, we're playing. And pretty soon, they're like sitting as they have that nostril, and they're looking up like, yes, this is so awesome. And then they'll just do that over and over again all day long. Then you're like, okay, well, you're too smart for your own good now. We're gonna go make it harder for you. So we go and we hide the samples all over the place, really random areas like on rocks, in logs. You put them in like right next to a stream. We put them in tree stumps, anywhere that's gonna make the dog really go and find it out. And they do every single time, and over again you'll do that until the dog starts searching areas on its own. And it gets a feel for what they want you to find and where they want you to find it. And it's actually the best time for the handler and the dog to bond as well, because you're learning to read the dog's changes in behaviors. You're looking for those tail wags. You're looking for the difference in the ears or the way the head is positioned. For instance, one of our dogs, Sadie Mae, she's the one in the field exercise photo. When she's searching for a sample, her tail just wags normal, back and forth, like the entire time. I think it's the only reason she's skinny. She just goes like this all day long. And then when she's on a sample, her tail hits a windmill. And she's just like, it is turning a circle like nobody's business. And you're like, wow, okay, she's on a scent, cool. But they all have different search patterns. Sadie turns big circles. Tucker on land, he's so lazy. He just goes back and forth all over everywhere. And then Dio, it's like hard to control him. He's like up on the logs, searching around. He's like jumping up on boulders. He's back on the ground. He's doing circles. He's doing whatever he wants. And then when he hits a sample, he's like, boom. He's like, gets low to the ground, and his like nose is fixed on it. He's pretty funny. So it's one of the ways you bond with a dog. It's one of the ways you learn all about them. And we're like, great, they're totally on the sample. This is perfect. And you can usually take them out in field study. Like for the ones on the moose, now they're working in the woods and they're doing all of that, having fun every day, picking up poop. When you put them on a boat, you're like, okay, well now what? Because one, the dog can't run at the sample for me, so their search pattern is completely gone. 2, I'm staring at the dog who's got his head hanging over the bow, trying to figure out what the heck his nose is doing. And 3, I have to figure out how to talk to Giles without turning around and distracting a dog. So we can find a sample and go 6 inches to the left. Well, what I think is 6 inches to the left, but assuming it's like 10 feet. Or 4 feet to the right. So it all goes out the window. But the main concept is we put it in a bowl. So we have stores and stores of poop. Actually, it's all in my freezer right next to my peas. So don't ever eat dinner at my house. But they all end up in a bowl. You float the bowl away. And then we drive the boat directly into the sample. And that's how you start the dog. So each poop gives off a scent cone. So like if you think of the poop as this projector in the front, this is actually one of the best examples. The whole theater at that point is your scent cone. So you're driving the boat straight down the middle of the seeds initially to get the dog used to looking at the water, used to smelling it without being able to move to it. And once they figure that out, you can move further out of the scent cone with the boat. And then instead of doing like big changes of behavior where on land they'll do like a 90-degree turn, Sadie's tail will start wagging like in that windmill like I mentioned, On the boat, she'll like jump to the left of the boat and her tail will start wagging. So that's our cue initially. Oh great, there's a poop in the water. And then we just drive back and forth till we find it in the water. And then what do we do with it?
So while we're out there too, we're maximizing the amount of time that we have on the water and really taking advantage of the fact that we have a research boat out there. And so we take information on the number and type of vessels and how they're, where they're located in relation to the whale. So we take about a half a mile around the group of animals that we're working with, and sometimes we'll even do a bee count if there's a separate group of whales in the vicinity with a totally different group of vessels around it. We'll take information, we'll call it a bee count. So just trying to capture the information. In fact, it's commensurate with the data that Soundwatch takes, so it's accumulating this larger data set for the general region. We take whale behavior also every half an hour, so who's there, if we can tell overall behavior, so are they traveling or foraging, resting or socializing, how fast are they going, are they directional pretty much, or are they kind of just all over the place. That information is similar to the way that other researchers in the vicinity take information on the killer whale behavior. We take photos when we can to be able to couple that with the sample if we do get a sample. Often if Dave from the Center for Whale Research is on the water as well, we'll call him and say, hey, who do we have? Because generally speaking, we're really far away from the whales. The whole point of the Conservation Canines program is to be as far away as possible while still maximizing the opportunity for Tucker to get that scent. And so we generally wait until the whole whale group goes by and then the fleet of boats go by, and we wait about 400 meters and then we start going in behind the whale and transecting behind them. So when people, when I got my job at the center, people were like, oh, who's this animal? And sending me photos. I was like, I don't know, send me a trailing edge of 400 meters and I might be able to help you, but I don't know. Dave? So then we collect date, time, distance from the whale, and Liz has set up a really awesome way of taking the data. It's an automatic track log that gets recorded as soon as we get on scene with the animals until the end of the day. And all of that's collected, so we have that information going back to 2008 on this study.
Okay, so there's 3 ways we find poop while we're out there, 3 main setups. We do a follow, a sneak, and a lateral. And it's all dependent on wind direction and how the whales are traveling, pretty much. So the one I have on here is a lateral. And the main idea is that the boat always has to be perpendicular to the wind. So, like if the whales, for instance, how many of you have been on the West Side whale watching already? Woo! Okay, great. So as the whales are going up Harrow, say from the left side of Land Bank all the way up to Limecombe, and the wind is blowing from Victoria or the other island across the water over there towards us, then we're going to be on the lateral. So we're going to be on the inshore side of them, which we know isn't the best place to be, but for us, sniffing poop, and like Giles said, we're usually well behind the whales anyways. That's where we need to be in order for Tucker or Dio to hit that sample. So we're also going to be back, and if there's a big ebb at that point in time, pretty much what happens is the whales will leave us a present. The wind blows it across towards the boat, and then we'll hit that scent cone. So what I've actually done is come up with just a little step-by-step, because it makes it a little easier to actually understand what we're doing. So here's the whales traveling up the middle of Harrow, right? Left side of Land Bank, going to Limecombe. The wind is blowing from the whales towards you guys on land. So we're traveling between the whales and land on that lateral. We're usually well outside of the Be Whale Wise and the guidelines, as well as the state and federal laws. Actually, 97% of our samples are collected outside that yardage. And then the water, we're on a ebb that's just pumping it down island towards Eagle Point, so we barely even have to move. And boom, there's our present. Yay, we're so excited. And this is what it does. So you get that scent cone, remember, from the projector all the way across the whole theater. That's your triangle scent cone. And in the middle, in that red area, that's where it's absolutely the strongest. So that's where Tucker or whatever dog we have is gonna make that jump to the left of the boat, or they're gonna do some sort of movement that means, okay, we're turning the boat in right there. And when we turn the boat in, we end up in that zigzag formation. As Tucker's leaning to the left, we're moving the boat to the left because it means we're too far to the right. And our main goal is to stay in that middle of the scent cone the entire time until we find it. Once we find it, this is the whole reason Tucker's out there. This is what his dream is. 1 second. So it's honestly, I'm pretty sure it's what he dreams about, and it's the whole reason he's out on the water. It's just to do that. And it's attached to a rope, which is attached to the boat, because the first time I ever went out on the boat, he threw his first ball over and it hit the water, and I was like, oh my God, are you kidding me? Now it's just floating away. And then I gave him my backup, and 2 seconds later, he throws it up in the air over the boat, and I was like, really? And I look at Giles, and she's like, yeah, he does that all the time. And I was like, no, this is not working for me.
And then it left me having to call on the radio, like, Tucker lost his ball, so you guys keep your eyes on him.
So the next day, we tied it to the boat, and we haven't had that issue. But it is his favorite thing to do, is he'll throw it over the boat. 9 times out of 10, it lands in the poop in the water. Which means when I pull it back on the boat, I'm like, everyone keep your mouth shut because it's really gross up here. It's really gross up here. So once we find it, how do you get it onto the boat?
So this is one of our volunteers. She's been out a couple times. She moved to Idaho and still writes to us regularly and posts things on our Facebook account saying, look, 4 years ago I was on Moja. So this is Annie. Our main volunteer, Sharon Grace, she is a champion. She, drop of a hat, last minute, oh my God, Sharon, can you go? And she's always like, tell me when to be there. And thank you for your hours and hours and hours of collecting and more recently for getting doused in whale poop. And just one-ups. We love it. Yeah, lots of hand sanitizer and mouthwash on board. So this is what happens when we get a sample. It's a little sped up. So she has a sample in that scoop. It gets passed back towards the back of the boat where we have an intern back there processing. So she's taking all of the excess water that gets collected in the beakers as well and pouring off the water. And just trying to get the fecal into a 50 ml tube.
Let's see. How many mils do we have? I'm thinking like 10, 15. Woo!
It's very exciting. It is exciting to get that much because actually when that spins down, that's going to be 3 to 5 mils, something like that. Sometimes all Tucker needs to smell a fecal in the water is a piece of fecal that's like the size of your fingernail. He can smell it, and sometimes that's all that's left. And we'll collect it, and we'll reward him for his awesome work. But it's not worth it. That little amount is not worth it for us to process and run through the lab. It costs how much per sample to?
For hormone?
Mm-hmm. $120 just for the hormone analysis per sample. It doesn't include DNA or any of the other things that we really need to be looking at too. So that gives you an idea. So we're just not even gonna, those small ones we don't collect anymore. So findings, we have over 600 samples since 2010, over 450 from 70 unique individuals, which is really great when our population is just in the low 80s. So that covers a large portion of the population. 170 of these are large enough for toxin measures, as well as some of the other analysis that gets done. So those have been done on 54 individuals. So Dr. Sam Wasser, the lead on this project's lab, has determined that when the whales first come in at the beginning of the season, where they've been out foraging on coastwide Chinook, that they come into the inland waters as healthy as they will be for the rest of the season. And just essentially that's a whole nother talk, that there's not enough Fraser River Chinook salmon here to keep their nutrition levels high. And so slowly, depending on how big the run is, their nutrition hormones are going to, their nutritional health is going to decline as the season goes on, depending on how much fish or how little fish is here. So one of the most recent findings, and this is a paper that's in prep, it's actually in review right now, which was really startling, was so Sam was able to determine pregnancy hormones from fecal, and in a captive situation, the only way you can do this is, well, they do it now with fecal as well, but they used to do it with blood, and clearly we can't do blood analysis from our wild killer whales. And so Sam's lab has perfected this way of looking at pregnancy hormones, and only 40% of pregnancies that we detect from females that we get fecal from, only 40% of those are end in a successful birth. 60% of them are lost before the calf is viable, so they're miscarried. And out of that, 24% are from late-term pregnancies, so that's a lot of nutrition and a lot of that female's energy has gone into growing that fetus. So that's really disturbing. So that you put that statistic on top of the out of the 40% that are successfully birthed, and you look at the loss within that population. So we know from older studies, Peter Alisiak studies showing 42% of calves don't make it their first year. So kind of putting those two stats together, it's really no wonder we're not seeing a population increase in these whales. Sam's in the process of correlating this with the fisheries abundance and date from when the female got pregnant. One of the interesting things that happened within this community, in 2013, the whales were missing from the inland waters more than half the amount of time that they normally are. They were somewhere getting pregnant in that summer 2013 and early 2014. They were somewhere out there, not in the inland water because they weren't seen. They were together enough to be getting pregnant a lot. And during that period, some of you may have been clued into some of the media that was going out. There was this big fervor, this big sad, I think, movement to say, oh, there's this great baby boom and the southern residents are out of the woods, their population is increasing. The problem with that is, number one, there were 11 animals born. Out of those 11 animals, 8 of them are still living. That's what we should be seeing in a regular year, 8+ babies within this southern resident, or 11+ babies in this community being born and living. We don't have that. Those years when those calves were conceived, and then the next 18 to 24 months were good salmon years, and I do this because they weren't good salmon years, they were higher than the previous years. And that's what allowed the southern resident females to hold their calves, gestate them, and birth them. And again, most of them, as far as we know, are living. Oh no, we've confirmed. Actually, we've confirmed all 8 of them now, just as of the end of last week. That's what we should be seeing all of the time. What we're concerned about now, these next couple of years are looking really, really bad for coastwide Chinook and for Fraser River Chinook. And so it'll be very interesting and I think sad to see what ends up coming out of this next 24 months with regard to successful births.
All right, so we mentioned one of the other things we look at, sorry, this is like the depressing part of the talk, is the toxins were one of the main reasons that these specific killer whales were listed as endangered. Through the fecal study, we're able to get all of that, the toxin levels that were listed out of the fecal. That was all Jessica London's work, and that paper was just recently published. But some of the main things, and I wanted to put this diagram up here, is because toxins are really bad, and they're legacy toxins, and they get into the waters, and we don't have a way of getting them out of the water once they're there. And it's hard for people to comprehend what exactly they're doing. So for instance, this diagram is showing how PCBs are getting into killer whales. They come from rainwater runoff that goes into the ditches or through the drain, like on the streets here, gets into the water, and scientists have found that it's not actually getting all the way to the sediment anymore. It's hitting the phytoplankton and the plankton, the very bottom of the food chain right away. So those levels right there are way higher than they ever expected them to be. And then it progresses from there. So you have the smolts and the feeder fish that are eating those. And then as they're growing up and they're eating more of the bottom of the food chain, they're just accumulating toxins. And what is happening is that they're stored in the fat layer. So as Giles said, Chinook are some of the fattiest salmon. So they're storing a lot of toxins in the fat, like in the meaty part of the fish. So when the killer whales are eating those salmon, they're automatically gaining all of the toxins that those salmon have. These killer whales need 100 to 200 pounds of Chinook a day, right?
Depending on, yeah, up to 300 if it's a lactating or gestating female.
So that's a lot of salmon that have already been completely loaded with toxins. These whales store it exactly the same way the salmon do. They store it in their blubber layer. So one of the things that was found as people started researching the toxins is that if the whales are fatty, then they're most likely not being affected by the toxins. But as soon as they start getting hungry, they start having to swim longer or search harder for that food, they're burning through the fat layer, the more toxins are getting released into their system. So toxins in general, they found to affect pregnancy, they found them to affect immune systems, the list goes on. There was actually a really cool study that showed regular people, if you're a salmon eater, on a regular basis, like 2-3 times a week, your toxin levels, specifically PBDEs, which are flame retardants, are way higher than an average vegetarian person. So just something for even people to consider in terms of killer whales, if that makes it easier for you. But just to know as well, if you're a seafood eater, you are gonna have toxins in your system. Sorry, a little depressing. However, some of the interesting things is DDT, which was banned in the 70s, is still found in huge levels in the killer whales. We've actually matched all of these toxin levels to the blubber layer biopsies that NOAA had. So we've validated all of the work. So K-pod and L-pod, which you normally frequent California to get that Sacramento River salmon, they have really high levels of DDT. J-pod, which spends a lot of their time in the Puget Sound, especially in the winter, swimming down to Seattle and hanging out around there, has really high levels of PCBs, which kind of makes sense when you think about all of the industrial runoff that Seattle has. You can also look up some other studies about the difference between coho and chum and how they're affected by rainwater even. When you have a heavy rain in Seattle hits into the streams, coho actually were found within 4 hours to come down with an illness from all the rainwater runoff. And then you have the flame retardants, which were actually found on the same levels for all 3 pods, which is really high and really detrimental for their health. One of the things we would love to actually look more into even is the toxin levels in the transient killer whales. So our goal for next year is to get the dogs trained on the transient fecal, get a lot of it collected so we can see a comparison between animals that, like killer whales specifically, that eat higher up the food chain and why the toxins aren't affecting them as much as they are the southern residents. If you have younger ones in this group, you might need to plug their ears for 2 seconds. These are some cool things, we'll go back to something fun, that you can find out of killer whale fecal. You have roundworms, actually, and we found, 20 to 30 of these in a poop one day where they'd eaten a salmon that had worms, and then the worms just grew in them, and there was one that was like almost 2 feet. It was crazy. It was so cool. So we collected it and gave it to other people to analyze it. We kind of have a thing for poop over here, in case you guys didn't notice. We really like the martini glass style of poop collector, so we thought we'd show you the different varieties, which is where the next 2 videos are coming from. So this one is because we're not quite sure what it was when we collected it. We showed it last year too, and somebody thought they knew, but it still hasn't quite been verified. So it's in the middle here.
There's a puffin in Cricket Bay and on channel
Can we pause that? So the main thing is that's Giles laughing, taking the video, and I'm the one having to hold it and figure out how to get it in those beakers. So. Yeah, she died. She, I think, was crying, laughing at me having to deal with this. We haven't figured out completely what it is. What we do know is this whale, because Tucker cued in on it, and we have a feeling it had to do with pregnancy or something out of that end rather than the back end. Well, the other back end. One of the things we wanted to show you is how we actually get it out of the water, what a normal quantity looks like, and hopefully normal quantity and the different colors it can be. This next one shows you what my favorite description of it is. Sorry, pancake lovers. But if you have a really hot griddle and you have your pancake batter, instead of making a perfect little circular pancake, you just kind of slosh your pancake batter all over your hot griddle. You end up with a really big blob and a couple little blobs and then a couple little blobs. That's what whale poop looks like on the water. And it's about the same texture if it's a healthy poop. And it ranges in color from tan to brown to green, red, orange. We had one person describe it as wonton soup with red chili flakes. So this would be, an example of how we scoop it out of the water. This one has an iceberg, if you get the drift.
Beautiful.
She gets very excited when I scoop poop. But it shows all the little pieces laying around, and it takes a little bit of technique to scoop it in. If you have questions, Sharon will tell you it's kind of like an egg on a water. It's like egg whites. You kind of got to scoop it in. And that's all we got for you. We do have the website up and running. It's conservationbiology.edu. And we do, like I said earlier, we post on Facebook all the time, whether it's about the killer whale study or our groups all over the U.S. And international. We're always on Facebook. So thank you very much for listening.
The marine labs?
No, we store our samples there, and it is a UW facility out there, but no, actually Liz stores them in her freezer now.
Yeah.
If that gets to be too, there's no room for peas at all, then there is a deep storage that we can use here, but then Sam's lab is actually on campus in Seattle.
And then where do you moor the boat?
Snug Harbor.
Snug Harbor, yeah.
Yes.
Do you test for pharmaceuticals?
We haven't yet, but we would like to. There's a grad student currently working on grants to test for exactly that, actually.
I was just told yesterday by a nurse at Swedish that Swedish, which is a major hospital system here in Seattle, that their policy is that if they have unused pharmaceuticals,
Right.
That they didn't use them all in the inpatient, that their policy is to flush them.
Right.
As opposed to put them into paper towel and put it in the
Yep.
trash. And she's trying to get the policy changed, but it is shocking in that with the University of Washington and San Juan Island, that is that major system's policy.
Yeah, the southern residents have actually been shown already through some breath analysis that Dr. Dave Bain did a number of years ago that they do have antibiotic-resistant bacteria in them, so they are picking that up from the water.
Yeah, I was actually going to say too, Sarah Potter, her master's work was actually looking at gut bacteria and antibiotic-resistant stuff. She found high levels of tetracycline.
Mm-hmm. Yeah.
How old do you have to be to a volunteer intern?
On that boat, you have to be about 18. Because it gets really crazy on that boat, and it could just be, it's kind of a university policy too, 18, and you have to sign a waiver even. But we want to see you in what, 10 years? 9 years? 5 years?
9.
9. I'm bad. Awesome.
Yes? In the back corner?
First off, to the sweet voice in the front row, you already have Dr. English. One is the presentation. I think I caught this in San Francisco. You guys presented their agreement society.
Jessica might have presented there. Yeah, was it more about the toxins?
I believe so.
Yeah,
Yeah.
so that's Jessica Lundeen, and she just finished her
Yeah.
PhD.
What you guys are doing. What is the expense to add something to the analysis that's pathology or toxins or pharmaceuticals? What would be the expense that you would need to have paid for in order to add new things to the list?
I think it depends on the analysis. What's the basic for the nutritional?
It's 120 just to run the hormones without including salary, anything else. Like it doesn't include the DNA, it doesn't include toxins. What we're saying is I think on average we're about $500 a sample to do toxins, DNA, salary, and the hormone.
And how many samples would you need to complete?
On a good year, we get 175 samples.
Okay. I can't do that.
It's a lot. Yeah, it's quite expensive. But the information we learn from it is just astounding.
Down here?
How are you funded?
We get mostly grants and private donations.
Can you donate through your website?
Yeah, absolutely. We have a donate button and we would welcome anything and everything. We also put out a calendar for conservation canines, and it comes out in the end of October actually. It's a year-round calendar put out by a professional photographer. The photos are unbelievable. And what you can do is earmark it for a specific study or interest as you donate for the calendar. Yes. That's what we're there for.
So we'll be out there if you guys have more questions.
Yes.
Yeah.
Yes.
Thank you.
Thank you.